All right, good morning, everybody. Once again, this is your teacher, Mr. Aguilar, here to talk about DNA extraction. Again, this will be another virtual lab brought to you by the University of Utah in their Learn Genetics program. You can actually visit that by going to learn.genetics.utah.edu for lots of different genetics-related materials, such as the one we will be going over today. So first thing we're going to do today is talk about DNA extraction. Why would we do it? What is it used for? How do we do it? Let's go ahead and get started, everybody. So we are going to be isolating some DNA from a human test subject. Again, why might this be something we would want to do? Genetic testing might be one aspect. We also have body identification. A little grim, but it is what it is. Also, analysis of forensic evidence. So again, anyone who's ever seen one of those CSI shows or any kind of a murder mystery, you're probably familiar with some of these DNA extraction techniques. And we know that DNA is a way every single person on the face of the earth has unique DNA. Now, identical twins technically have the exact same DNA, although as they get older with age and stuff, telomeres and other things might end up being a little bit different. So that is another subject for another time. But essentially, DNA is an exact blueprint, copy, fingerprint that is unique to every individual on Earth. Granted, mothers, fathers, next of kin will have similarities in their DNA, but everything is still unique. So if you have someone's DNA, you can basically match it up with them, and that's how they use it for forensic evidence and body identification. All right, so typically, DNA extraction is going to be essentially the first step in a longer process. It's very important because in order to use and study the DNA, we need it to be purified. Originally, DNA, as we all know, was found inside the nucleus of a cell. It is also found inside the mitochondria. But when it's inside the nucleus of the cell, it is wrapped around histones, there's cytoplasm, there's all kinds of organelles and other debris that surround it. And if we're going to study it, we need to clean it and isolate it. And that's what we're going to be doing here in this lab. So we got a cell, there's our nucleus, and there's our DNA wrapped around the chromosomes, or included in the chromosomes and wrapped around the histones. I'm sorry. So we got histones there, and we got DNA as we see it. We see the base pairs, we see the double-stranded helical structure, all these good things. So where do we begin? Well, first we need some cells, right? DNA is found inside the nucleus of cells, so that's what we're going to need. Usually, you can get it from hair, you can get it from skin, you can get it from nails, you can get it from anywhere. However, the insides of the cheek, very, very quick and easy place, very painless place to get DNA. We lose thousands of cells in our mouths every day. Very simple process. Basically, we're going to get a little swab, kind of like a Q-tip. Swabby, swabby, boom, got the cells we need. So this is what we're going to need to do. We're going to need to collect some cheek cells. Then we will need to burst them open to release the DNA. Then we have to separate the DNA from all the protein and debris and everything else. And finally, we are going to isolate our concentrated DNA. So as I said before, here's some different things we're going to be using. Let's go ahead and get that going. So we have our water bath, a centrifuge, micropipetters, buccal swabs, sample tubes, lysis solution, concentrated salt, resuspension buffer, and ethanol. Ethanol is like alcohol, and we also have isopropyl alcohol. All right, let's go ahead and get started. I'm going to take our buccal swab here, and let's go ahead and get some cheek cells. Swabby, swabby. Got what we need. Yo. All right, so our cheek cells we should have on our swab. There we can see some of the cells right over there. Now we're going to place the swab into an Eppendorf tube so that we can start to burst them open and isolate that DNA that we so desperately need. So cut off the little swabby part. And now we're going to take a lysis solution. Now the lysis solution is what's going to burst open those cells and release that DNA. So let's take our micropipette. We filled it with the lysis solution. Bloop, and pop it right into our Eppendorf tube with our swab containing our cheek cells with DNA inside. Now we're going to go ahead and take our tube and put it into a nice warm water bath. Get it all comfy and relaxed. Okay, there's our cell. Uh-oh, we're breaking it open, breaking it open. There's our chromosome. We're going to have to break that dude open too, aren't we? Bloom. So now we are bursting open our cells. And the Lysis solution, again, as I said, has two important ingredients. It has a detergent and a proteinase K. The detergent disrupts the cell membrane and the nuclear envelope, which causes the cell to burst open and release the DNA. However, DNA is still wrapped very tightly around proteins called histones, so the proteinase K cuts apart the histones to free the DNA. So it's basically, the proteinase is something that breaks proteins down. So it's wrapped around these protein histones. The proteinase K is going to break down the histones, releasing the DNA. Okay, so now we've released our DNA. It's still filled with a whole bunch of junk, but hey, we're on the right track. Okay. Now at this point, we are going to take our concentrated salt solution. We're going to get our micropipetter, take our concentrated salt, and then we will put it into our tube. So what this is going to do is it's going to cause all the proteins and debris in the tube to start to kind of clump together, which will essentially leave the DNA free to float, and it'll be much easier for us to separate it. So as we can see, we have all the clumping of protein and debris and the free-floating DNA. That's good. Now what we're going to do is we're going to take a microcentrifuge, which, I don't know, you can almost think of it like as a clothes dryer or something, or one of those things at the, what do you call it? One of those rides at the carnival where you go in the tube along the wall and then it spins really fast and you get stuck along the wall. It's kind of like that. So the protein and the debris weigh a whole lot more than the DNA, and they're all clumped together. Remember, DNA is tiny. It's on the nanometer scale. Proteins and all that other stuff are much larger. So what we're going to do is we're going to spin it really, really fast to get all those big proteins and everything all stuck at the bottom, leaving our pure DNA floating in our liquid. Now, when we use a microcentrifuge, we also need to put a tube on the other side that basically weighs it out. Otherwise, it's going to be uneven, and it spins so fast that it could not explode, but it could break and it could shatter the tube. Just imagine if you've ever had like a pair of tennis shoes in the dryer. It kind of messes it up. It makes it all uneven. The dryer starts walking all over the place. Similar situation. Let's go ahead and put that top back on our centrifuge there. Alrighty, so as I said, we spun it super duper quickly. All that protein and debris is all down there in the bottom, and now we have our free DNA floating in the liquid. So this is great. We're one step closer to the process of purifying and extracting the DNA that we so desperately need. So we're going to go ahead and take that liquid out of there because that's where our DNA is going to be. We can go ahead there and we will put our liquid containing the DNA in a special tube. And now we can use some isopropyl alcohol. Now we're going to add that to the tube, take some isopropyl alcohol and put it in our tube. Now DNA is not soluble in isopropyl alcohol, which is a good thing. That's going to help us to separate it out. It's going to precipitate, to use a vocabulary word there. So there we go. Now we have our DNA kind of clumped together and separated out. You can even see it. See those kind of white streaks? That is DNA. So you're going to vert the tube several times to make sure everything gets mixed well. And again, like I said, since DNA is not soluble in the isopropyl alcohol solution, it will begin to clump and to precipitate out. This is good. So once again, now that we have our clumpy DNA, we're going to use our handy dandy microcentrifuge. We've got to make sure we even it out. There you go. Pop that top. Let's spin it up and let's see what we get. And if you said DNA is going to clump at the bottom, ding, 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 you've won. Wonderful. All right, now we don't need that liquid anymore, so we're going to extract it. Once we have it removed, the DNA is allowed to dry. You can redissolve it in a solution of your choice, store it in the freezer for many years, or you can move on to your next experiment. So guys, that is how you collect, isolate, clean, and purify DNA. It's actually a fairly simple process. It seems like it'd be way more complicated than that, but it really isn't. So wonderful. You guys can actually do this at home. I put this up last week. I will put up a link to it again. You can extract DNA from pretty much anything. It's really easy to extract it from soft, squishy fruit like strawberries or mango or berries. So any of you guys who want to do some extra credit, I will put up a link that shows you how to extract DNA using ingredients and things you have at your very house. You can go ahead and do it, take a video of it, take a picture of your extracted DNA, and I will give you guys bonus credit. All right, everybody. Next thing we will be doing, we'll be going over the DNA electrophoresis. That is how you separate strands of DNA. So when we're looking for specific things, we can extract the DNA in terms of size. So I'll show you how to do that next. Uh, alrighty, it's been real.